Hold your breath. Not for a minute, but for the next half hour. Because we are going down, and the rules you live by are coming apart with every meter. At 200 meters, the colors die. Red goes first, then orange, until the ocean locks itself into a dim blue dusk. By 1,000 meters, sunlight is gone entirely, and it will never come back. And yet, every layer we pass has life in it, life that looks progressively less like it belongs to this planet. There is a fish down there that absorbs 99.5% of the light that touches it. A jellyfish carrying tentacles longer than a blue whale. A worm with no mouth that dissolves bone in acid. None of this is decoration. Every terrifying thing you are about to see is a solution, an answer to a specific problem that depth creates. By the time we reach the bottom, you won't see monsters anymore. You'll see engineering. Let's descend. Start at the surface. The water here is warm, lit, and organized entirely around photosynthesis. The euphotic zone, from the surface down to roughly 200 meters, holds the vast majority of ocean life. Nearly every fish that has ever appeared in a nature documentary lives here. Reef fish, tuna, sea turtles, whale sharks. It represents less than 4% of the ocean's total volume, but produces almost all its primary food. This is the world where surface biology makes sense, where bodies are shaped by rules that have governed animal design for hundreds of millions of years. Those rules begin with light, and light isn't a single thing. It's a spectrum of wavelengths, and water absorbs each wavelength at a different rate. The principle is consistent. Longer wavelengths carry less energy and are absorbed first. Red light, the longest visible wavelength, around 700 nanometers, is largely gone within the first 10 meters in clear, open ocean water. Take a red fish, photograph it below 10 meters, and it appears gray. There is no red light remaining to reflect. Orange disappears by roughly 13 to 14 meters, yellow by around 50, green by 90, blue-green by 150. By 200 meters, only the narrowest band of cold blue light persists, and even that is fading. This gradient isn't just atmospheric. It's the structural cause of everything you're about to see. Every creature below the sunlit zone inherited a world where the visual language of surface biology no longer functions. Every species that crossed that boundary had to answer new questions. The answers are the creatures we are going to meet. 200 meters. The twilight zone begins. Formerly called the mesopelagic zone, the twilight zone extends from 200 to roughly 1,000 meters. The light here is dim, directional, and almost entirely blue-green. What matters most isn't what you can see, but what sees you. A predator scanning upward from 250 meters can still detect the silhouette of a prey animal outlined against the faint glow from the surface. That silhouette is a death sentence in a water column where there is nowhere to hide and nothing to retreat into. Every creature in this zone is under the same selective pressure. Eliminate the shadow that marks you as prey. 3 engineering solutions evolved independently. They share nothing except the problem they answer. The first, absorbing every photon that strikes you, was taken to its extreme by the Pacific black dragon, among 16 deep-sea fish species studied in detail by Alexander Davis at Duke University and Karen Osborne at the Smithsonian's National Museum of Natural History. In 2020, their team published measurements from 16 species of deep-sea fishes collected from Monterey Bay and the Gulf of Mexico, spanning 7 entirely unrelated taxonomic orders, each reflecting less than 0.5% of visible light. Ultra-black means absorbing more than 99.5% of incoming light. The darkest individual measured, a dreamer anglerfish in the genus Anirides, returned only 0.044%, placing it on par with the blackest known animals on Earth, including the birds of paradise of Papua New Guinea. The mechanism is structural. In ordinary dark-skinned fish, Melanosomes, pigment-containing organelles, are separated by unpigmented gaps within the skin. In ultra-black species, those gaps are gone. A continuous layer of melanosomes sits packed in the outermost dermis with no interruptions. The melanosomes themselves are larger and more elongated than those in other dark vertebrates, not arranged in any ordered crystal pattern, but randomly packed so densely that light scattered by one melanosome is immediately intercepted by its neighbor. The optical path lengthens. The melanin absorbs. Almost nothing returns. Osborn described the arrangement as melanosomes packed into the skin cells like a tiny gumball machine, where all of the gumballs are of just the right size and shape to trap light within the machine. The paper's sighting distance models found that ultra-black skin reduces a visual predator's detection range by more than 6-fold compared to fish with ordinary dark pigmentation. The constraint it solves, bioluminescent light from a predator's searchlight or from nearby organisms, would scatter off ordinary scales and outline the fish against the dark water. The Pacific black dragon absorbs that light before it returns. The fish isn't just dark, it's optically absent. The second solution is addition, manufacturing exactly the light needed to cancel your own shadow. The hatchetfish, genus Argyropellicus, took this path. Its flanks are silvered, reflecting ambient light laterally. Along its belly runs a line of ventral photophores, tiny light organs pointed straight down, emitting blue light, wavelength matched to the downwelling radiance from the surface. The Hui twilight zone creature feature describes the effect concisely. Counter-illumination. Tiny light organs on its belly produce a glow, exactly matching the light filtering down. That glow is actively controlled. Laboratory experiments confirmed that hatchetfish activate their photophores in response to overhead illumination and dim them when ambient light drops. A reference system built into the fish, photophores oriented toward the eye itself rather than outward, continuously samples the ambient light and calibrates the belly output accordingly. The result is a fish that replaces its own silhouette with a signature indistinguishable from background illumination to anything looking up from below. Hatchetfish run both strategies simultaneously. Silvered sides handle lateral visibility. Ventral photophores handle the view from below. One body, two exposure problems, two solutions running in parallel. The trade-off is precision. The spectral output of those photophores is largely fixed by blue optical filters built into their structure, meaning intensity can vary, but the exact angular distribution can't be perfectly controlled. The system isn't flawless. It's good enough to have persisted for millions of years. The third solution is removal. Make the body transparent. Glass squids, family Cranchiidae, roughly 60 described species, have bodies so clear that the only externally visible structures are their large, opaque eyes and a cigar-shaped digestive gland. Mbari's animal page describes the condition directly. Because they are so clear, usually the only visible parts are their large, opaque eyes and a cigar-shaped digestive gland. The gland is oriented vertically, minimizing its shadow when seen from below. Small photophores at its ventral tip and under the eyes emit light at the same intensity as the downwelling background, cancelling the shadow cast by the one part the body can't make clear. The mantle cavity contains ammonium-rich fluid that provides neutral buoyancy, allowing the squid to hang motionless in the water column, invisible to what is below, and not moving in ways that might draw attention to what remains. 3 strategies: maximum absorption, active emission, material transparency, all built around the same constraint. Each carries a cost. Ultra-black skin can't adapt to bioluminescent sources positioned laterally. Counter-illumination fails if the predator looks from the side rather than directly below. Transparency fails around every organ that can't be made clear. The twilight zone isn't an arms race with a winner. It's an ecology of engineering compromises that has been running without interruption since before mammals existed. 1,000 meters, and the last traceable photon of solar light disappears. The bathypelagic zone, the midnight zone, is, in terms of raw volume, the largest habitat on Earth. No surface light arrives. No counter-illumination strategy makes sense against a backdrop that is already fully black. What animals here carry instead is light they generate themselves. The 17-year survey of water column life, conducted by Severine Martini and Stephen Haddock of Mbari, tracking observations from the surface down to 3,900 meters off the California coast, published in Scientific Reports in 2017, found that roughly 76% of all macroscopic organisms encountered had bioluminescent capability. That proportion held remarkably constant across depth. Bioluminescence in the midnight zone isn't a speciality, it's the default. The functions are diverse: luring prey, signaling mates, defensive startle displays, and one application that runs in an unexpected direction. In bioluminescent dinoflagellates, the microscopic plankton responsible for many ocean flashes, mechanical disturbance triggers a light burst. When a zooplankton grazes on a dinoflagellate, the disturbance produces a flash that illuminates the grazer, potentially attracting a larger predator to the site. The dinoflagellate is consumed regardless. The flash draws something that may eat what ate it. Among larger deep-sea creatures, the same logic appears to extend. Some animals release bioluminescent clouds when attacked, creating visible disturbances that may redirect incoming threats. The ocean runs on layered predation, and bioluminescence threads through all of it. The anglerfish's esker is the most vivid expression of bioluminescence as a hunting architecture. Female ceratioid anglerfishes develop a modified rostral spine, the esker, a fleshy, glowing bulb suspended on a filament above the mouth. The esker glows because of luminous bacteria from the Vibrionaceae family living inside it. Baker and colleagues confirmed in eLife in 2019 that anglerfishes acquire these bacteria from ambient seawater during early development. They aren't inherited. Each generation rebuilds the symbiosis from scratch. The bacteria are genetically reduced, stripped down to what is needed to produce light and survive in the lure. In total darkness, with almost nothing to eat, the esker manufactures the one signal prey can't resist: a light source. The mouth is below it. The anglerfish doesn't pursue, it waits. The constraint, absolute darkness combined with extreme food scarcity, produced a living lamp attached to a jaw. Every expansion of the anglerfish's body architecture follows from that lamp. A stomach capable of accommodating prey larger than its own body, jaws hinged to open wider than its head, a metabolism calibrated for months between meals. As depth increases through the midnight zone, even the nature of vision changes. Research on deep-sea vision describes a fundamental transition. In the upper mesopelagic, downwelling light creates an extended visual scene. By the bathypelagic zone, that extended scene is gone, replaced entirely by discrete bioluminescent point sources, individual flashes visible against a black background. Deep-sea eyes at this depth are tuned to detect single points of light rather than to resolve shapes. No eyes take that logic further than those belonging to the giant and colossal squids. Both Architeuthis dux and Mesonicoteuthis hamiltoni possess eyes measuring approximately 27 centimeters in diameter, based on the best available specimens. Dan Eric Nielsen and colleagues documented a giant squid eye of this size in Current Biology in 2012, with a pupil measuring roughly 9 centimeters across. The most complete adult colossal squid ever recovered, a 495-kilogram specimen now held at the Museum of New Zealand, Te Papa Tongarewa in Wellington, shows comparable dimensions. Every other living animal has smaller eyes by a significant margin. These measurements are based on the best available specimens. Colossal squid maximum size remains a subject of ongoing research, with much of the data extrapolated from beaks and mantle remains rather than intact adults. Nielsen's team modeled what an eye of that scale could detect below 600 meters. At 27 centimeters, the eye can identify large bioluminescent disturbances from more than 120 meters away. At shallower depths, a smaller eye works equally well. The size advantage only materializes when detecting something very large at significant range in near complete darkness. Sperm whales hunt giant squid. As a sperm whale moves through bioluminescent plankton-rich water, it disturbs those plankton, generating a glowing bow wave ahead of its body, a signature detectable well before the whale arrives in striking distance. Nielsen's analysis suggests the squid's eye evolved specifically to read this signal. As the eyes to see summary of his research describes it, this is basically a burglar alarm. The squid doesn't need to see the whale directly. It reads the whale's approach from the light the whale disturbs, from 120 meters away, with enough time to flee. By the deepest reaches of the midnight zone, point source vision becomes unreliable for many species. The lateral line becomes the primary sensory tool, a system of mechanoreceptive hair cells called neuromasts, running in canals along the body and head, registering pressure waves and water displacement directly through the skin. In cavefish populations that lose functional eyes over generations, neuromast density increases and canal morphology enlarges. The same pattern appears in deep-sea fish with reduced or absent eyes. Where light can't reach, pressure wave detection provides the sensory picture instead. At some point in the descent, depth doesn't just change what you can see, it replaces the sense entirely. 3,000 meters, the abyssal plane. Pressure at 4,000 meters stands at approximately 400 atmospheres, 400 times the weight of air pressing down on a square centimeter at the surface. The primary threat of that pressure isn't mechanical crushing. Most deep-sea bodies are water-based and nearly incompressible. The threat is molecular. High hydrostatic pressure disrupts enzyme function and protein folding, breaking the metabolic machinery that sustains cellular life. The abyssal zone covers roughly 1/3 of Earth's seafloor, extending from about 3,000 to 6,500 meters, and it's cold, near freezing, and completely dark. Food does not grow here, it falls. Marine snow is the technical term for what drifts downward from the productive surface waters. Fragments of dead plankton, fecal pellets, mucus aggregates, the shed scales and remains of organisms that lived in the zones above. Individual small particles sink at a meter or two per day. Aggregates can sink at 100 to 200 meters per day, meaning a particle leaving the surface can take 3 weeks to 2 months to reach 4,000 meters. Most of it's consumed or remineralized by bacteria and zooplankton on the way down. Only a small fraction survives the full descent. The abyssal economy runs on what the zones above failed to eat. Scarcity at depth produces one of the ocean's counterintuitive features, gigantism. Giant isopods, genus Bathynomus, relatives of the terrestrial woodlouse, are the most photographed example. Bathynomus giganteus reaches a typical length of 19 to 36 centimeters based on specimens examined in published Gulf of Mexico population studies. Supergiant species in the genus have been documented up to 50 centimeters. A body plan essentially unchanged for over 160 million years, distributed across the deep Atlantic at depths from a few hundred to more than 2,000 meters. Giant isopods survive more than 5 years without food in captivity, gorging when food appears and dropping their metabolism between arrivals. Cold temperatures slow metabolic rate, enabling longer lifespans and potentially larger maximum sizes. Reduced predation at depth removes evolutionary constraints on body mass. A larger body buffers against irregular feast and famine cycles. None of these hypotheses is settled. All may operate simultaneously. The most dramatic food event in the abyss isn't snow, it's gravity. When a large whale dies, a blue whale can weigh up to 150,000 kilograms, The carcass sinks over days, settling on the sea floor as a whale fall. The succession has been described in 4 stages. In the first, mobile scavengers, sharks, hagfish, sleeper sharks, strip soft tissue over months to more than a year. In the second, enrichment opportunists, including polychaete worms and crustaceans, colonize the skeleton and surrounding sediment. In the third and most productive stage, anaerobic bacteria begin breaking down lipids locked inside the bones, generating hydrogen sulfide that feeds chemosynthetic communities analogous to those at hydrothermal vents. This sulfophilic stage can last decades. In the fourth, the bare skeleton persists as hard substrate for suspension feeders. A single large whale fall can sustain this full succession for up to 100 years. It's in the sulfophilic stage that Osedax appears. Robert Vrienhoek, Shana Goffredi, and Greg Rouse of Mbari first collected the worm in 2002 from the bones of a grey whale carcass at approximately 3,000 meters in Monterey Canyon. They formally described it in Science in 2004. The most remarkable feature is structural. Adult females have no mouth, no gut, and no anus. Instead, Osedax females have root-like posterior structures that penetrate the bone. Martin Tresgueras and colleagues confirmed in a 2013 Proceedings of the Royal Society B paper that these roots express vacuolar H-ATPase, a proton pump, alongside carbonic anhydrase, the enzyme that hydrates metabolic carbon dioxide into hydrogen ions and bicarbonate. The proton pump drives those hydrogen ions through the root epithelium directly onto the bone surface. The acid dissolves the hydroxyapatite matrix, exposing the collagen and lipids trapped inside. Intracellular bacteria of the order Oceanospirales, living in specialized cells within the roots, then break down those liberated compounds and transfer energy to the worm through a pathway that bypasses ingestion entirely. The Smithsonian Ocean Portal summarizes it without flinching. Osedax secrete an acid from their skin that dissolves bone, freeing up the fat and protein trapped inside. Then, symbiotic bacteria living in the worm's bodies digest the fat and protein. No mouth, no gut, acid applied to a surface, bacteria processing the result. A complete redesign of how an animal can eat, built from a constraint that produces no organic food except locked inside bone.
Vrienhoek's 2008 paper confirmed what the discovery team had observed. With one known exception among described species, every Osedax female carries inside the gelatinous tube surrounding her trunk a colony of microscopic males. These males are paedomorphic, permanently larval, never developing mouths, digestive systems, or any structure beyond what delivers sperm. Nature Citable describes the female carrying hundreds of males within her tube. Deep-sea News puts the size ratio at roughly 100,000 to 1 in favor of the female. The males aren't organisms in any full metabolic sense. They are the worm's reproductive mechanism, miniaturized to near invisibility and metabolically negligible. The female's body reorganized entirely around extracting nutrients from bone. The male's body reorganized entirely around delivering sperm. Both extremes answer the same constraint. Whale falls are rare, spatially unpredictable events, and each one must be colonized by reproductive females as quickly as possible. Minimize male energy consumption. Maximize female presence. Run reproduction through the female's tube. The worm with no mouth, dissolving skeletons with acid, housing microscopic male harems. Every one of those traits is a functional solution, nothing more and nothing less. 6,000 meters. The hadal zone begins, and the trenches represent less than 1% of the ocean floor's total area, while constituting a biologically distinct region separated from the abyssal plain above by pressure differentials that function as hard limits on which body plans can persist. The limiting factor is molecular. Paul Yancey and colleagues, writing in PNAS in 2014, documented that trimethylamine oxide, TMAO, increases approximately linearly in teleost fish muscle with depth, increasing approximately linearly between roughly 900 and 4,850 meters depth. TMAO is a piezolyte. It stabilizes protein structure under pressure, counteracting the tendency of high hydrostatic force to disrupt enzyme conformation and cellular function. Extrapolating that linear increase, bony fish would reach isosmotic conditions, where TMAO-driven osmolarity equals seawater, somewhere between 8,200 and 8,400 meters. Beyond that depth, the biochemical cost of maintaining sufficient TMAO appears to exceed what vertebrate physiology can sustain. This is why no large, fast predators exist in the deepest trenches. Their enzymes stop functioning. Their cell membranes destabilize. Speed, complex musculature, and high metabolic throughput require biochemical stability that pressure eventually defeats, regardless of body size or evolutionary ancestry. What survives instead is small, soft, and metabolically slow. Pseudoliparis swirae, the Mariana snailfish, was formerly described from specimens collected between 6,900 and 8,300 meters. Subsequent expeditions have produced observations at approximately 8,336 meters, making it the deepest confirmed vertebrate on record. Translucent, roughly 30 centimeters in length, scaleless, and gelatinous in consistency, a body plan that minimizes structural pressure differentials between the animal's tissues and the surrounding water. There is nothing rigid inside it to implode. Amphipods, Hirondalia gigas specifically, function as the hadal zone's primary scavengers, documented at the full depth of the Mariana Trench. Crustacean-jointed, small and soft, they process the organic material that collects at the bottom of the trench. Trench topography concentrates food. The V-shaped walls funnel marine snow toward the deepest points, making the floor of a trench relatively food-rich compared to the flat abyssal plain surrounding it. Below 6 kilometers, the engineering currency is no longer anatomical. A molecule that prevents proteins from collapsing. Body plans that eliminate rigidity. Metabolic rates slow enough that the trickle of arriving food is sufficient.
The adaptation language at this depth is written in concentrations, not in claws or eyes. The Challenger Deep is the deepest measured point in the Mariana Trench, measured at over 36,000 feet, with survey-to-survey variance producing figures differing by more than 100 meters, depending on methodology. It sits roughly 330 kilometers southwest of Guam. The trench around it runs 1,580 miles long. The first scientific measurement of that depth was made in 1875 by the crew of HMS Challenger, the British research vessel conducting what became the first global oceanographic survey. The ship carried 181 miles of Italian hemp rope for sounding. At station 225, they lowered a weighted line and measured approximately 4,475 fathoms, about 8,184 meters by modern conversion. On at least some attempts, accounts suggest the rope ran out before the weight reached bottom, meaning the crew understood they had found something their equipment couldn't fully confirm. The first humans to see the floor of the Challenger Deep arrived on January 23, 1960. Jacques Piccard, son of the bathyscaph's inventor Auguste Piccard, and Lieutenant Don Walsh of the United States Navy, descended in the vessel Trieste. The bathyscaphe used gasoline for buoyancy, nearly incompressible under extreme pressure, and carried iron-shot ballast released magnetically when the crew needed to ascend. The descent took approximately 4 hours and 47 minutes. During the dive, an outer viewport cracked. The sound was abrupt, and Picard noted it. The crack was non-catastrophic, and they continued down. The temperature inside the sphere dropped toward freezing. They spent 20 minutes on the seafloor. What they found wasn't the barren desert most surface scientists had assumed. A flatfish-like creature moved across the pale sediment. The identification remains debated. Some researchers have since suggested it may have been a sea cucumber seen at an unusual angle. The floor itself was soft, diatomaceous ooze, not empty. Before ascending, Walsh sounded 6 tones on the underwater communications device. Picard dropped 2 tons of iron shot. The Trieste began to rise. 2 men in a steel sphere, gasoline keeping them afloat and iron weights holding them down, descended to where the pressure was approximately 1,100 times what exists at sea level. They did in under 5 hours what evolution has been doing without interruption in the surrounding water for hundreds of millions of years. What evolution placed there, at the absolute bottom, wasn't a different kind of biology. It was the same logic carried to its most extreme expression. Minimize structure, maximize biochemical tolerance, wait for food, and don't move unless necessary. 3 creatures didn't appear in this descent, and they deserve naming. Each one answers a constraint this script didn't reach. The vampire squid, Vampiroteuthis infernalis, is neither a squid nor an octopus, but a separate cephalopod lineage, occupying the oxygen minimum zone between roughly 600 and 900 meters, where dissolved oxygen is too scarce for most active predators to function. Rather than hunting, it collects marine snow on long, sticky filaments, feeding on detritus. An animal shaped like a predator that behaves like a recycler. The oxygen minimum zone is the specific constraint that produced it, and nothing else would have. The barrel-eye fish, Macropinna microstoma, lives between 600 and 800 meters with a transparent, fluid-filled head shield enclosing 2 tubular eyes. Mbari researchers published in 2009 that those eyes can rotate. They aren't fixed upward as scientists had assumed for decades. The fish can reorient them forward to track prey as it feeds, while the transparent dome protects the eyes and admits light from nearly every direction simultaneously. A moving window, built into the head. Siphonophores, specifically Prior dubia, may reach 40 to 50 meters in total length, placing them among the longest organisms on the planet. They aren't a single animal. Each structure in the chain is a distinct individual called a zooid, performing a specialized function: propulsion, feeding, defense, reproduction. Together, they operate as one coordinated organism. The constraint they answer isn't one of depth alone, but of scale. At some point, the individual body plan reaches its evolutionary ceiling, and colonial organization becomes more efficient than any single animal could be. If you want a second descent, into those 3 and into the others waiting at the edges of this one, leave a comment. We will go. We started at the surface, where everything made sense, where animals looked the way animals are supposed to look. And then the light died, and the pressure climbed, and life started writing stranger and stranger answers to harder and harder questions. Every creature you saw tonight was a receipt. The black dragon's darkness is a receipt for a world with nowhere to hide. The anglerfish's lure is a receipt for an ocean with no food. The worm's acid is a receipt for the only meal that ever falls this far. The deep isn't cruel, it's honest. It shows you exactly what survival costs when you strip away everything easy. And the next time you stand at the edge of the ocean, remember, beneath you, right now, there are 6 miles of solutions, and every single one of them is still working.